All right, folks, welcome back to the Serious Angler podcast, and welcome back to another edition of the Real Biology here at Serious Angler. Always with myself, Mr. Bailey Egbert, and Mr. Stephen Bardin. But we have a special guest, and Steve, why don't you introduce the folks to our special guest for today?
Yeah, today's a special episode because we have Dr. Cynthia Fox Holt, a true expert in a multitude of things, lives in the DFW area, works for Texas Parks and Wildlife as a inland fisheries biologist, but also she's had experience in Puerto Rico. We're going to chat about that because I'm going to Puerto Rico next year, so I want some
Really?
travel tips.
Okay.
She's going to answer one of the questions that we all have about those big black spots that show up on our largemouth and smallmouth. And we're here together, which is the first time I've had a guest. I'm going to call it in studio. We'll call it partially in studio.
Do I hear some fighter jets in the background?
Yeah.
Probably.
That's pretty cool.
We're not in the Middle East. We're in West Texas. We're actually at a kids camp. So Dr. Holt and I are instructors for a camp called the Bass Brigade, which is part of the Texas Brigade's youth leadership camps. This is my 12th year. It's probably like your
My second.
second year. I was going to give her 5. But yeah,
I didn't get wrangled in until last year.
Yeah, so this camp is available for students 13 to 17 years old. They come and hang out on a West Texas ranch with Texas Parks and Wildlife biologists like Dr. Holt. All of my txprolake staff volunteers. Gary Klein comes and teaches some fishing technique and sonar and all kinds of cool things with him.
We also have several high school biology teachers that come out and work with the students, sharing some of their fishing knowledge with them, just general fish nerds.
Right, a bunch of fish heads. And right now, the students and most of the instructors are down at the stream, the Jim Ned Creek, and they are backpack electrofishing. They're pulling a seine. They're collecting little bugs with kick nets. They're talking about watersheds, doing like a riparian walk. So they're like in the water collecting fish and IDing them and all those things right now. They'll come back up here this afternoon and we're going to do some fishing skills, some fly fishing skills. They're here all week, and tomorrow they'll do water quality. They'll jump on the electrofishing boat. They're going to experience what it's like to be a biologist and learn for a week what we do. If anybody's interested in the camp, I'm sure Bailey will put the link in the description below. It's going to be texasbrigades.org, and I'm sure there's like a click through to Bass Brigade. I used to be the camp coordinator, but at this point, that's been passed on. We're on Glenn Chancellor now, and one of our West Texas technicians is now the camp coordinator. So we're here for the week. I thought this was a cool opportunity to have an in studio guest with Dr. Fox Holt, and her and I have worked on a couple things. So it's gonna be a fun episode, Bailey.
Yeah, and you perfectly lined it up for me, Steve, because I was just going to ask, like, if there's somebody listening, whether their kid or themselves wanted to get involved, how to do that. Yeah, we'll definitely link that down below. And I guess where you're at in Texas with everything going on is all okay in your part of Texas with all the chaos and all the.
Thanks for asking that, Bailey. I would say Central to South Texas experienced some devastating flooding with a tropical storm kind of remnants that came across the Pacific, across Mexico, and just hovered over parts of Texas. And Central Texas, I mean, there were some reports of like 20-inch plus rainfall, in one single day. And our camp was not affected. We're about 100 miles north and just a little bit west. We're not affected, but man, we can't say enough about the impact that storm had on this Central and South Texas community and the whole state of Texas. And our hearts go out to it. A lot of our game wardens are there. We usually have a game warden here. Probably isn't going to be able to attend.
Won't be able to. No, they were deployed to Central Texas to help with search and rescue, and
Yep.
And they're out there. Feels like 24/7. We're getting information from them, so it's just so devastating, so sad.
And 20 inches of rainfall, that's about 10 inches less than we get annually in those areas. So that's, almost a year's worth of rainfall in one day. It's a big deal.
Yeah. videos and photos just that were being seen is just it's incredible in regards to just one like from the magnitude of how fast and how much came down. It was
Correct.
just insane to watch from the outside, but I guess in a half glass full, it's cool to see all the support that's going down that way from organizations, brands, things like that that are stepping up, not just throwing funds, but also people that are coming from all over
Right.
the country to come down and help. And yeah, sad deal. And we'll try to link some things down below as well if anybody would like to go and try to help and support
Cool.
from
And
that
we
end.
did, we were actually reached out to by our Hupper administration letting us know because obviously the game wardens were sent because they have lots of search and rescue capabilities and training. Most of our fishery staff do not. Occasionally we do get deployed to help, but we were told, and so this is kind of a general public thing, that right now they have the people they need.
Good.
So, don't just hop in the car and drive down there because ultimately you would be adding to the chaos rather than helping. So, really do some research and figure out what the best way to support is because a lot of us, we want to jump in, we want to be, we want to be boots on the ground, but sometimes there's too many boots. And so the best thing you can do is send some cases of water or something like that. So definitely check those links and make sure that you're putting your efforts in the right place because we all want to help.
Yeah,
Well said.
love that. Well, hopefully we can bring some positive light in regards to some fish talk now. So hopefully some more positive conversation and things like that while the boots on the ground are helping where it's needed. But regardless, welcome to the show. Welcome to the real biology here, and we got some cool stuff we're going to talk about that. A lot of people always, I think, even myself included, have been curious of like why bass have these blotchy spots on them, and we're going to talk about that, and I'm sure a bunch more in this episode. But yeah, with that, Steve, kick us off.
Well, I think that's a great place to start. That's one of the first projects you and I got to work on together.
Yep.
You were years ahead of me, but you did reach out to me eventually, and I appreciate that from the Major League Fishing side. We were glad we got to help
Yeah,
in a
that was
small
awesome.
part. But start at the beginning. Don't answer where the black spots are yet.
Okay.
Let's start at the beginning. What is the research behind figuring this out? What is the rules?
Okay, so we have several different sections of it. The research is primarily being led by the federal agency USGS, U.S. Geological Survey. It's based out of their research facility in Leetown, West Virginia. It's the Eastern Ecological Science Center. I happen to know the PI on the project. He's a good friend of mine. We went to college together. And he was contacted by local anglers in the New York sort of northeast area of the United States about seeing the black spots on smallmouth bass.
Mhm.
And they were seeing them more frequently in the fish they were catching. And then there was a fish kill, don't remember why, but there was a fish kill, and most of the fish in that had blotchy spots on them. So they were like, is this associated? So that's actually what led to the impetus
Yeah.
of the project.
And that was like 2018-ish or so?
Actually, more like 2012.
Okay,
The
2012.
publication came out in 2018.
Perfect. Okay.
So the research started in 2012.
So
Was
you're
that
go ahead, Ben.
was that Oneida Lake by chance? If you
No,
recall
it
the
was
lake.
a river. It was a river.
Oh, okay.
Yeah. Okay, so the USGS team started looking into because of the fish kill.
Hmm.
Your role in Texas, you're so far away from New York. How did you get? Yes, you went to school, but
Yes.
what was your role?
Yes. So because he works for a federal agency, I can't remember the name of the act. It's a basically it's a Congress legislation that does not allow federal government to openly ask the public for information.
Okay.
So he reached out to me because I am a state biologist, and state biologists are not covered under that act. So we are allowed to reach out to our public and ask for information. So as a biologist in one of the largest metroplexes in the country, I am connected with almost 100,000 anglers through a few Facebook groups, and then just in person, I see them at outreach
Okay.
events that we work at. And so I contacted them because some of these people are fishing 7 days a week. And so I thought that will get us hands on tons of fish.
Yeah, what were y'all looking for specifically?
So we were looking for any largemouth bass, smallmouth bass, black bass in general, actually, that might have those irregular blotches on the external part of their body. And when we got really excited was actually when we saw that some Guadalupe bass
Right.
had the blotches. And so that really kind of ramped up Texas's desire to understand what was going on. And Texas anglers are phenomenal. We had over 1,000 entries in about 8 months.
So you're just on social media, you're meeting anglers, and you're saying, if you see these,
Yes.
contact Dr. Holt because I want to know.
Send me the photos.
Okay.
Send me the water body where you caught it.
Okay.
And if you can tell me the length and the weight of the fish, if it looked healthy, that would be great.
Right.
And of course, we kept all that information private. There were no fishing spots were made public to anybody, but it was an outrageous number of fish that we saw, which is funny because as biologists, we see hundreds of fish every year, and we never really noticed it.
Right.
Never noticed it until we started looking for it.
Right.
And then it was everywhere.
Yep.
Everywhere. We can go, there are some lakes in our district we can go to, and 1 out of every 4 fish is going to be blotchy.
Okay.
It's insane.
Well, and this is why I had to have Dr. Fox Holt on the show. This is what real biology is. This is a group of scientists that say we need help from the public, reach out to anglers. Have you seen blotches on bass? And you get 1,000 yeses within 8 months.
Yes. And because the Texas effort was so fabulous and so fruitful, we were actually able to reach out to other states and some Canadian provinces and Mexican provinces that anglers provided images to either their state agency or directly to me. And so we ended up having reports, so images from anglers reported from 32 states in about a year and a half.
Okay, so in my mind, this is what I'm hearing you say. In New York, we had this little issue. We had a fish kill. We had some questions about these blotches. Then you said, "Hey, Texas anglers, do you see it?" And so you confirmed it in Texas. Now we have like a distribution.
Yes.
And now you have 32 states, Canada, Mexico. It's a North American issue.
Yes.
So it's not regional. It's not your specific fishery. It's all of North America, basically.
Yes.
Okay.
Where largemouth bass, where black bass are.
Where black bass are.
So we did get a report from the state of Washington, but through the images it looked like blotchy bass, but we were not able to confirm it through the biological testing of the tissue.
Yep.
So as of right now, Texas is about as far west as it goes confirmed.
Okay. Let's pause for a second and make sure the audience understands exactly what we're talking about. Because a largemouth or a black bass in general can have natural coloration patterns on their body that we would call blotches.
Yes.
So in science, it's kind of this weird thing. Like below the lateral line on a largemouth, you have blotches, which are pigmentations that come from the scales that are slightly darker. They can be present or they cannot be present depending on watercolor, clarity, time of year, temperature, so many variables. They will change color even throughout the day. You catch a bass, you put it in your live well, it was white when you caught it, it comes out really green and spotted or blotchy an hour later. What we're talking about, though, is like the big black tattoo bass.
Ink-like blotches. That's the terminology that we tried to use to separate it from the standard blotch of a bass typical coloration. Is it looks like an ink-like spot on the scales of the fish or on the mouth or the fins. It actually occurs most commonly on the fins and the lips.
Gotcha.
So it looks like they're wearing lipstick.
So how many entries did you get that you were like, I don't see anything, this is just normal skin?
Several. Because especially for largemouth bass, they actually have a dark spot on the posterior of their operculum, and I got so many of those.
That means the gill covering.
Oh, sorry. Sorry, my bad.
Oh no, please. This is perfect because I like to have, and Steve is great at dumbing. This is why I'm here. I'm the
layman's
dummy in
terms.
the group always, so I need to have it in simpleton terms, but it's good to have what they're actually called,
Yep.
'cause
Okay.
that's
Okay.
how we educate.
Okay. But yes, so many anglers send in photos and they're like, hey, is this blotchy? And we're like, no, that's actually just a natural coloration. But thank you for your mission. Keep looking. And almost, they were, oh, okay, cool. So a lot of people were able to learn a lot. They also, misidentified blotchy for a couple of other, illnesses that can occur on fish. Black spot is one of them, but it's a very different illness that I don't know absolutely nothing about. So please don't ask me.
That's okay.
But yeah, and one other distinctive characteristic of the blotchy bass ink-like spots is that if you touch it, it will slough off. So like if you rub it with your finger, or we were actually using buccal swabs, that was one positive of COVID-19, unfortunately. But if you want to think silver lining, it made non-lethal data collection on fish very easy, because the same swabs they use to test for COVID are the swabs that we used to collect Blotchy Bass Syndrome skin cells from fish. And they will, when you wipe it with the swab, the skin cells come off, so the black spot goes away.
Interesting.
It scared me the first time that I did it, because I was like, oh, what did I do? And so I contacted the biologist, Clay, at USGS, and he says, oh yeah, that's how you know you got the sample right, is it came off. That's, okay.
So phase 1 was this, let's get all the pictures. Let's find out distribution. Phase 2 is what you're talking about now, where you actually physically collected specimens, and then you wipe the buccal swab to get skin cells.
Yes, from both wild caught and captive populations. Bass Pro Shops, Cabela's, were very instrumental in allowing USGS to get some controlled samples, because most of those fish are wild caught fish that then go into the tanks. And so when they would see the blotches on them, they will actually carry blotches sort of year round. Whereas your wild populations, the blotches will come and go based on a few different environmental factors that we're not exactly sure how they interact with each other. But you can see, based on reporting from the anglers, the highest percentage of reports came from November to April. April was actually the peak of reporting. So right during bass spawning season.
Right.
So might have a reproductive hormone component to it. We haven't been able to determine that yet. But with the captive fish in Bass Pro tanks and Cabela's tanks, those fish would keep the blotches kind of year round. So they were able to track more of, how it operated on the fish, even though it was a captive population. So that's what allowed us to really, because I don't know how much you know about viruses. I know very little, but what I've learned is that you have to build the virus genome to be able to describe it. And so they were able to do that using the Bass Pro and Cabela's fish. So then when we went out and collected wild specimens and sent them those swabs, they could then compare it to those to determine if it was the same virus or a different virus.
So she's letting the cat out of the bag here. That's okay.
Sorry.
That's okay. This is how it works. We get so excited about it, we just want to talk the science.
Just goes.
So you've got the black spots, you start the buccal swab, you got a control population. So then the next step was to map the genome.
Yep.
Okay, so what did we figure out? What is causing, or what do all the black spot fish have?
Well, I can't say it's all the blackfish. There is no definitive always. But most anglers, and they're probably thinking this right now as they're listening to your podcast, saying, well, it's just because of their diet, or it's just UV, which even as fishery scientists, prior to this study, that's what I was telling people when they would send photos. Yeah, it's probably just their diet. It's probably UV. I'm sure they're fine. No worries. Release them. Nothing. But turns out that bass, just like humans, just like lots of other animals, carry bacteria and viruses on the outside of their bodies.
Right.
But it normally doesn't make them sick.
Okay.
However, Black basses have a type of virus that's called an adenovirus. It's a specific family of viruses.
Okay.
I can't tell you much more about it other than it is typically species specific. So there's a specific virus for one species of fish, a different species of fish. We do know that it's not transmissible to humans.
Okay.
Big point, not transmissible to humans or pets.
Okay.
And they carry the virus, and then when they get stressed, so spawning, temperature, oxygen, water quality, something like that, that starts the immune response. And the blotch, the ink-like spot that forms, is actually the immune response to the stressor.
Okay.
So it's basically if you have a bug bite
Yep.
and you get the red inflammation and the itching, that's essentially what the ink-like spot is on fish. It is that reaction to the stressor.
Case closed.
That's what it is.
It's an immune response to a stressor.
Yep. And it could be any stressor. We haven't been able to determine a specific stressor.
So in this, let me summarize so that I remember it. So we have the Bass Pro Shop fish that are in a controlled environment. You map the genome of the skin cells that are coming off the blotch, and then you're able to collect wild caught fish. How many states, like how many people participate in this part?
Probably 20 states provided samples.
Okay, so 20 states from across the country provide samples, and those blotch samples were matched genomically. So it's the same virus. It's not like 150 different ones. It is a black bass specific, smallmouth and largemouth and Guadalupe's.
We have, so the only ones we've confirmed individual viruses are largemouth, which there might actually be a difference with the debate about Florida bass versus northern bass.
Okay.
There might be a specific genomic virus between those two species, as well as a genetically separate virus for smallmouth and a genetically separate virus for shoal bass. Those are the only ones they have been able to confirm so far.
So let me think about this now. Smallmouth and largemouth are different species. We all know that. We all agree to that. Bailey, you're good with that? Okay. So if I have a smallmouth with a black blotch, that's an immune response to a virus that's on the smallmouth. And it, say it again,
Adenovirus.
Adenovirus on a smallmouth. But that same adenovirus in the water comes into contact with a largemouth, however it gets there. It's not going to give that same immune response.
No, sir.
Because it's a different species.
It's a different virus. Yes.
So you've mapped 3 of them so far.
Yes.
Largemouth, which would be northern largemouth, or?
They've mapped both.
Oh.
And there is a slight difference between the northern and the Florida, because we now know that genetically the northern and the Florida bass are different.
Right.
And so if you have this, so it's very scientific, we all know DNA is made up of the 4 bases. You can have one base change that will lead to a phenotypic change, which is largemouth to smallmouth. Or you can have a base change that doesn't lead to phenotypic change, but it leads to a genetic change. So Florida northern, the adenovirus is doing that.
Okay.
So it is, they're all adenoviruses, but they are very species specific.
All right. So the black blotch is an immune response to an adenovirus that is linked to the species. I'm going to ask you a question I don't expect an answer for.
Okay.
What about hybrids?
So they actually were able to map the genome.
Okay.
And it is a mix of the 2.
Oh my goodness. I don't even know where to go from here.
So viruses are crazy.
Viruses are crazy.
But they have one thing that I really want people to know is the viruses have essentially been around as long as the bass have. This is not something new. Humans have not, it's not a pollution. Humans have not caused it.
Okay.
This is a completely natural thing.
And at this point also, it's not impactful. It's not.
We are not sure.
Okay.
So that's the next step. We actually have sort of 2 next steps, because unfortunately the anglers submitting photos took off so much farther and so much faster than we expected that it wasn't until about halfway through the project with me and with the Angler Atlas that, which I'm kind of jumping ahead there, but with both projects, it happened so quickly that we didn't think through. We had distribution, but we weren't able to determine prevalence.
Okay.
So it is completely possible that every fish that has this stress response that's having those black blotches is going to be perfectly healthy and fine and live a super long life and everything's great. It might also be possible that they're all dying and ending up at the bottom of the lake and we don't know it because when they get sick, as most of us know, they don't actively hunt, so they don't actively bite lures, and so anglers are not as prone to catching them. So we're not really sure if it is negatively impacting populations, but because we know that it is becoming more prevalent and it is, of course, a virus that is related to stressors, and stressors are becoming more and more frequent in everyday life, this is why we need to continue to study this to see how it will affect populations, especially when you consider the fact that it is species specific. Right. And the Guadalupe bass is a very small population in a very specific area of the state.
Right.
We did have some of those fish submitted with images that looked like they had blotchy bass. So this is why, as a biologist in Texas, and as part of TPWD, we do want to see
more fish.
What's going on.
Okay, so I want to think about the bass pro shop fish for a second.
Okay.
Because the bass pro shop fish are in a controlled environment, and we can kind of watch them over time, but they'll have this stress response, this blotch, and then they'll continue to live.
Yep.
So, at least in a captive environment, we feel like they're okay.
Yes.
Okay.
Yes.
Man, Bailey, how's that for a nerd beginning to an episode?
That's perfect right there. I mean, there's been so many different things that I've heard on it where it's like, oh, it's just a sign of a healthy fishery. Oh, it's a sign that that fish, you hear it all the time. They have the black spots because they're around a bunch of other fish. That's how they get them. And then it's, no, this is the actual reason.
Yep. My favorite one was, oh, it's because they're eating crayfish.
Yep.
That was the most common response that I got, was it was because they were eating crayfish.
And now that you've said that, in the comments, somebody's going to say, what about the red lips? So do you want to tackle? Nope.
Nope.
Yeah. I always, because a lot of times I see the black spots as we're coming out of winter into that spawn.
Yep.
And that's the same time that we do see bass with red lips. And I've also heard the same thing about the red lips. Like, it's because they're eating crayfish. And I always say, well, in my mind, once again, this may or may not be true. In my mind, the causation of that would be temperature. They need blood to go to certain parts of the body. They're starting to forage at a greater rate because they're getting ready to spawn. We see similar things with stress response in a live well. Like, if you leave a fish in a live well, you'll have red fins, red lips, because that's blood being pushed to certain areas of the body.
Well, and they're bumping into the walls of your live well, and they are actively feeding. They typically feed on sunfish, which are spiny. They typically come from behind, so they're probably catching those spines on the lips, and so that's leading to that inflammation. Maybe they're using, they're bumping other fish to protect their nest and their eggs. So that's always what I thought the red lips were. But I also used to think that blotchy bass was related to diet or sun. So who knows?
And I think this is the cool part about science is you have two scientists that live and breathe this every day that are saying, I have an opinion, I have a thought on what I think this could be, but it's not based on thousands of hours of observing and trying to answer this question. It's just an environmental observation. This is what those fish should be doing right now. So this is what we think it is. And at some point, somebody, maybe because of a fish kill or because of they're looking into live well mortality or whatever, somebody will research it. And that's what's cool about this blotchy thing. You mentioned Angler Atlas. And I want to go down that route a little bit more. I think, Bailey, do you have any further questions on the virus?
No, I think we pretty much answered that.
I do have one other step. So I mentioned the one direction of the prevalence. And the reason we weren't able to get at that is because anglers were only reporting blotchy fish. They weren't reporting all of the fish. And that's what leads into the Angler Atlas
Perfect.
thing. But one other side to that is we are not sure how the virus spreads.
Fair.
So, because oddly enough, within Texas Parks and Wildlife, we don't see blotchy bass virus in any of our hatchery fish. None of them. And we raise the sharelunkers. We looked through the history of the sharelunker photo database, and there was not a single blotchy bass in all of those entries. Not
Okay.
one.
I'm shocked by this.
We were too.
So
we don't. So obviously, even though the idea is that they do carry it, there is a point where they can be stressed and not show the immune response clearly, but we don't know why. We don't know how it spreads. And so one of the subjects that we are going to try to tackle in future years, if we can get the grant funding to do it, is to do a laboratory study with control and blotchy fish with different types of contact, just water, fish to fish, human hand to fish, because one really unique fish that was seen on an eastern coastal state was it was a smallmouth bass, and when they electrofished it and they pulled it up and they looked at it, there was actually a thumbprint of a blotch in the mouth. So,
so that
seems like it came from the angler to the fish. So that's one direction that the research is heading. Okay. But then, thanks to Angler Atlas, so that's My Catch Angler Atlas. It is an app that is free to download, and it allows you to submit your catches. They use it to collect a very broad scale study information, and they were willing to work with USGS on this. They developed sort of an angling tournament,
right.
And anglers could sign up for free, and when they would go fishing, they would open the app, start their fishing trip, and then every fish they caught, they would submit that fish. They would put an image of that fish on their app, and then that gave us a better picture of prevalence. However, that was much more effective in the northeastern states than it was in the southern states, because the southern states
don't have cell phone service.
No, they don't want to show their hunting holes. They didn't trust it.
That's
but true. I'm just kidding. No, a lot of times it was one of those things where they would forget.
Gotcha.
They would get out, they would be fishing, and then 10 fish in, they catch a blotchy fish. Oh yeah, I was supposed to be submitting this. And then they start. Gotcha. Well, then they may not catch another fish. So it looks like 100% of the fish they caught were blotchy when really they weren't.
Gotcha.
So, but the Angler's Atlas app was a phenomenal part
of
this study. Is that
tournament still going on?
No.
Okay.
So that tournament was for one full year. We started it in March of 2022.
Okay.
And then continued it through March of 2023.
Gotcha.
So that we made sure to get that full spawning season. And it was a perfect thing with the fish. We saw the rise in the numbers in the spring, went down in the summer, tanked in August. You saw very few fish with it in August, except up in the northern states.
Okay.
And then started to come back up, and the tournament ended. And so many organizations, Bass Pro Shops, Cabela's, they offered gift cards. AFTCO, yes, offered gift cards and prizes for people that participated. And I was blown away by the number of entries that we got.
See, and that's, in my opinion, that's the power of anglers.
Yes.
As biologists, you said it yourself. Like, you go out and you electrofish. Let's say you electrofish. You're in DFW, so Louisville Lake, right?
Yes.
You electrofish Louisville. You got 20 stations you're going to do. You're 5 minutes per station. I don't know exactly how you'll do it.
That's exactly. We have 24 stations, but they're all 5 minutes. Yeah.
So in that, you do that at night. You may see one.
Maybe.
And it's nighttime. So you may not even get it.
And it barely has a blotch. It'll be like a pinhead blotch.
But Angler Atlas, running for a year, you know, let's say they just have 1,000 anglers, but they have several thousand anglers. They're going to get a lot of examples, even from Louisville, if those anglers participate, where you would miss them. And that's the power of anglers. Like as scientists, the more partnerships we create like that, the better our data collection is.
Yep.
And I think, you've answered a great question about blotchy bass, but I think the moral is when we can partner together, we speed our science up a lot.
Absolutely. And the perfect example of that is Belton Lake. So most Texans know Belton Lake is where you go if you want to catch smallmouth.
Right.
Texoma and Lake Fork, yes, are fabulous, but Belton is, as a transplant, Belton is the smallmouth bass lake in Texas. Our Waco team surveyed that reservoir. They did 24 5-minute stations.
Mm-hmm.
They caught tons of smallmouth bass. None of them were blotchy, but yet we had 200 or 300 anglers that submitted photos of blotchy smallmouth bass from Lake Belton.
Okay, I got another question. You just sparked it, and you may not be able to answer this, and that's completely okay.
Okay.
Smallmouth bass aren't from Texas.
Correct.
Blotchy bass would follow species line based on the adenomviruses that are species specific. How did that virus, that blotch, that's specific to smallmouth, get to Belton Lake?
It came with a fish, because those fish would have been from an area where smallmouth bass are native.
Right.
So they would have been carrying the virus on them
from a hatchery.
But
our hatchery
or possibly from a wild population.
Right. But our hatchery doesn't have them.
No.
So Texas raises like 4.5, 5 million Florida bass a year, roughly. And the brood stock, we don't see blotches on them.
Correct.
But that doesn't mean that they don't have the virus.
Correct.
What that means is that they don't have a stress response to show you a blotch.
They don't have a strong enough stress response to show you a blotch.
Amazing. All right. So from the Angler Atlas data, can you tell me what is the smallest fish that we saw with a blotch?
The smallest fish we saw was about 12 inches.
About 12 inches. So have you ever seen one
So I did see one in one of my ponds in the city of Dallas that was about 6 inches, and there is no way you would have convinced me that this fish was not blotchy. The blotches were there. We wiped it with the buccal swab. The blotch came off. We sent that swab to USGS, and it was negative for adenomvirus.
Wow. Okay.
So it seems like it only presents in adult fish.
Okay. Huh.
So that blotch, or what I saw as a blotch, may have been a stress response to the electrofishing. Or the handling. Yes.
Absolutely.
We see that happen quite frequently. So it was about 12 inches was the smallest fish that we saw it on. That said, there is some evidence that bluegill and a few sucker species and possibly even channel catfish have a similar adenomvirus that causes the same immune response, the black blotches.
And I've seen it on green sunfish.
Yep.
So,
yep.
Okay. What about the largest fish?
I want to say maybe around 4 to 5 pounds.
Okay. Because you said none of the sharelunkers.
None of the sharelunkers.
Sharelunker in Texas, of course, it's during a spawning window, January to the end of March. But that would be a 13-pounder bigger fish that's going to go to the hatchery. We haven't seen one of those. But you're saying 4 to 5 pounds. There's some gray area there. That's okay. That's okay.
And then actually, I did personally see an image of a smallmouth bass that was about, I think he told me it was 6 pounds.
Wow.
And it had a blotch. Very distinct.
The angler wasn't a guy named Andy, right?
No.
Okay, because he's a liar. Bailey, have you caught these with the black blotches before?
Yeah, largemouth and smallmouth. More up here in New York, I've caught more smallmouth that have them. Literally spotted cow looking ones. We call them Dalmatians when we catch them. But yeah, I mean, we don't see a lot, but we definitely see them in various of our fisheries.
Any big fish or just normal?
The one that had 30 spots on. I even have a whole photo. I have a photo of it and everything. It was about a 5.5 pound smallmouth.
Amazing.
Yep. That is cool.
That's a big one. That's a big one to have.
And
spots.
New York is actually where it was first observed by an angler. It was in the Hudson River in New York.
And Bailey is in New York, and he constantly is looking for what can an angler group do to participate. So I'm sure we could have some ideas for him on different things they could do if we want to continue this blotchy bass.
Absolutely. I'm still getting images from anglers on the regular, not just from Texas, but from other states. And I have a nice file. I put everything in, collecting as much data as I can so that it stays within the public eye, really. So, want to keep it in the public eye, keep people thinking about it so that, if there's a big enough push, then maybe we will be able to get the funding to do the additional research that is needed.
Yeah, I kind of want to talk about that funding part. And you may not know, on a single sample, what is it costing to run just one single buccal swab to say this is a virus, this is not a virus?
About $20.
About $20 per sample. And how many samples do we need? Where do we need to take this?
Well, at this point, we really we need samples from black bass that are not Florida, northern, smallmouth, and shoal. We need samples from Guadalupe, from spotted, from Chattahoochee, Alabama. We need it from those fish because those are the fish that we don't have the genome mapped yet. So
that is more difficult.
Yes.
Yeah, that is more difficult, especially because some of those have to have genetic verification on top. Yes. So then you're looking at $20 to run your buccal, plus you're looking at like $14 to run your genetics.
Plus purchasing the equipment, getting it to the anglers because it has to be shipped out. There is a specific, process that you have to go through. The good news, again, silver lining to COVID, they were able to develop an mRNA solution. So just essentially a solution that will protect the genetic material that can stay at room temperature. So it doesn't have to be frozen. It doesn't even have to be refrigerated, and it'll last for about 6 months.
So at the collection part of your study, y'all were actually using anglers to collect the samples as well.
Yes.
Oh, I missed that. Okay, so tell me a little bit about that.
So we had a few anglers in Texas and a few other states that said, hey, I have some in a private pond or at this community pond where I like to go fishing. I've seen it. What can I do? And we asked them if they would be willing to accept some swabs and then send them back. We sent them prepaid envelopes, swabs. Everything was labeled for them. They just had to write down the information about the fish and where they caught it and their name so that we knew, who the samples were coming from, and then they would send those in. So that was actually how we were able to get a few samples from some water bodies, private water bodies, because as Parks and Wildlife, we are not allowed to work on private water bodies. And some of those pond owners were willing to provide full history of stocking, the hatchery where they receive their fish, all, things like that. And then it really allowed us to kind of narrow in on, it's not just public waters. It's not just water bodies that Parks and Wildlife have stocked, because that was a question. Is it something that's coming out of the hatcheries because we're overproducing, we're crowding, something to that effect? We were able to prove, no, it's not. It is a completely naturally occurring virus.
Amazing.
And so
it's all over North America.
It's all over East North America.
All over North America.
Texas East.
Right,
Texas north to Canada and east.
Gotcha. All right, let's switch gears a little bit. Okay. This project is one of the reasons you and I got so close and got to know each other, but you do a lot in the DFW area that is also kind of part of my passion project with small impoundments. So I'd like to talk about that for a little bit, because we think of Texas and we think of Fork. Now people think of Ivy, we think of Rayburn, we think of Toledo Bend, and you have some major reservoirs as well.
I do.
Louisville was on display this year.
Yes.
But there's another part of being an inland fisheries biologist, which is not just reservoir management.
Yes.
And it's small impoundment, community management. Tell me a little bit about that track. Number one, did you see yourself going into that?
Absolutely.
Okay, so this has been part of the vision.
Yes. When I decided to become a freshwater fisheries biologist, which I did not start there. I started with sharks like most kids.
Right.
But when I decided to become a freshwater fisheries biologist, and I myself learned how to fish, which I did not learn until later in life. I was well into my third degree before I started enjoying fishing as an activity. But I realized early on when I was in college and I would work with young children, and to see them catch that first fish that they actually cast the line out, set the hook, reel it in. That was one of the most amazing experiences for me. And to get to pass that on to children that don't know that that's an option, that they don't know that careers like this exist, to see that light bulb go off or to see that bright smile is the epitome of a good day for me.
Right.
And so, reservoirs are awesome, but they can oftentimes be difficult for a lot of anglers to access because they don't always have shoreline access, and not everyone has access to a boat or a fishing pier that goes way out, kind of like what you see at the coast. So I wanted to focus, and I started to focus on those smaller impoundments so that I could have a bigger impact with more people. And honestly, the biggest thing that I have found as I've worked in the Dallas-Fort Worth Metroplex is that usually they don't need fish, they need habitat.
Yep.
That's where it goes, because most inner city ponds, community ponds, they're not built with creating a quality fishery in mind. They want a pretty water feature, something that's great to look at, which they are. I don't care if they are a bulkheaded fishbowl or a vegetated, oasis. They are all pretty to look at. Water is just pretty in general. But when people see water, they want to fish.
Right.
And so, and unfortunately, I don't think there's a state agency in North America, and I may be wrong, but I don't think there's a state agency that can produce enough fish to stock every water body every year all the time.
Correct.
And so my job as an urban fisheries biologist is to figure out how to provide quality fisheries with the resources that we have. And so 9 times out of 10, it's a habitat issue because you don't have the right area for fish to spawn and reproduce, so you don't have a bunch of new babies coming into the population, or you don't have an area for those babies to hide from the predators. And then you also maybe be missing something that is building the base of the food chain, like invertebrates, phytoplankton, paraphyte, and things like that. And so even though I'm a fisheries biologist, I'm very much becoming a fish habitat biologist.
Yes, I love it.
And so I just recently have completed 2 major projects with cities, and then I've got 2 more on the books that are coming up where we're not even talking fish yet. We are just talking pond construction and possible habitat opportunities. So gravel for spawning beds. So because bass and sunfish are nest builders, the males are going to go in, they're going to build their nest, females are going to lay the eggs, and then the males are going to protect it so that the babies survive. Well, next to those gravel beds, we typically like to put vegetation, because then when the babies are born and, when they hatch and they start swimming on their own, they'll head straight to that vegetation that will provide them with food and protection from predators. And so I will go to a pond, and the first thing I do is I look at the entire shoreline. And then if I can, I put my boat in and I'll look at the bottom. I'll see what the depth bathymetry is, so see what the contours look like, figure out where we can put these habitats where it will protect the fish, but also provide a quality fishery for anglers, because we try to look at the pond as a whole. So we all know that bass and sunfish and catfish love structure. So rock pilings, trees, the things like mossback fish attractors, PVC fish cubes, crappie condos with bamboo, which those don't typically work in ponds because they're too tall, but things like that. So when I look at a pond, I work with the city to show them, okay, you have a great pond, now let's make it a great fishery. Let's put some gravel here, let's plant some vegetation here. I work really closely with several local aquatic plant producers, and they help me determine what plants will be best for that pond based on water, substrate. our goals, control, because of course everybody worries about, oh, are the plants going to get out of control and then we're not going to be able to fish. Well, they are able to help me select plants that either won't get out of control or are very easily controlled without harming what's in the pond. And so I'm learning a lot about aquatic plants here lately.
I love this. You said so much there that I need to unpack. First, you started out like everybody, I want to work on sharks. So you wanted to go the marine biology path.
I did.
And you learned that bass are way more aggressive. So I would rather work on bass is what you decided.
Yep.
You grew up in kind of the mid.
I grew up in Middle Tennessee.
Okay.
No ocean anywhere.
So you just saw this on TV?
Yes. Okay. Yes. And every TV show and every book I read, the only people who were researching sharks were people with PhDs.
Okay.
So at 11 years old, I told my mom, I'm going to get a PhD in marine biology.
Perfect. And you stuck to it all the way up until you met
Dr. Quentin Fontenot and Dr. Elise Ferrara at Nicholls State University in Thibodaux, Louisiana.
And what was the tipping point there?
I visited because I was struggling to find a graduate program that would suit me, that I could be able to study sharks, because at the time a lot of people were studying the dead zone in the Gulf. Mm-hmm. And so most research was going to that. And I went, Fisheries World Small, so my professor in undergrad had just taken on a PhD student that had finished his master's with Dr. Elise Ferrara. So he put me in contact with them. They invited me down to see the university, and they were awesome. They basically told me, they said, look, we have a satellite campus that is on the coast that you can go to, but you're going to have, very little support staff. All of your professors are going to be here. There's very little funding. You're going to have to get your own grants. You are going to be essentially on your own.
Right.
Or you can stay here, switch to freshwater, which management is management. Mm-hmm. And we have funding. We have lab space. You can pick your project and go for that. And so I was just telling the story to someone earlier today. Prior to that, I had only been fishing 3 times in my life, and which is weird considering I lived 20 minutes west of Del Hollow Lake, where the world record smallmouth bass was caught. But I had never seen a largemouth bass that didn't have the dark coloring. Mm-hmm. I'd never seen one. So Quentin takes me out, and we set a gill net, and I'm pulling it in, and I see this fish that has a white belly and a lime green body.
Right.
It's got a big mouth. The underside of it is very white, flexible, flimsy, and I'm staring at it for what felt like forever. And Quentin said, Cynthia, what's wrong? And I said, Quentin, what is this? He says, that's a largemouth bass.
Right.
I was like, no, it's not. That was my introduction to largemouth bass sampling.
Amazing.
And so I was actually doing a study on yellow bass, so temperate basses. That was what I was working on. But I was setting gill nets, so they are non-specific. They catch everything. Right. And so that was the first time that I realized that fish could change color to adapt to their environment. And I just became fascinated. And after that, I was all about the freshwater fish. Fish. And I loved management, and I wanted to introduce the public to it. So I just continued with largemouth bass because bass is king.
Right. Absolutely.
That's what people want to fish for. So
we agree with that. So from I still have so many questions. These 3 fishing trips you did as a child, I presume.
Mm-hmm. Yes.
Who took you? Where'd you go? How old were you? Roughly.
Okay. So I actually can tell you exactly for all 3.
Perfect.
So my first fishing trip was devastating.
Okay.
Because where I grew up, there was a gentleman who had a farm pond, and he would allow the community kids. He would provide us cane poles. He had a worm box that he raised the earthworms in, and we could come and we could get them. He had little measuring boards, so he used us to cull his fish. So he would let us come up and fish, and anything for the sunfish of a certain size and bass at a certain size, he had it marked. Certain size you can take home, other size you put back in.
Okay.
So my brother and I had gone up and we had fished and we took home a mess of sunfish.
How old were we?
I was 4. My brother was 10. And we get home. My mom, who had fished and hunted
Right.
as a teenager, cleans the fish and she's cooking them. Because, I mean, they were sunfish, but we were proud,
You
so she
caught
was
them.
cooking them.
Yeah, absolutely.
She started to have an anaphylactic reaction to the fish.
Oh, no.
So we ended up in the emergency room.
Okay.
The very first time I ever went fishing, I sent my mother to the emergency room.
Okay. And do you know why this happened?
She just developed the allergy later in life to the fish. And she said, of course you would select a career that I'm allergic to. Of course you would.
Absolutely. So that's the first trip.
So that was the first trip.
Okay.
The second trip, I was 16, and I was fishing with my high school boyfriend.
Okay.
He was older. He was fishing in a bass tournament, and I was just on the boat. I wasn't actually fishing, but I was just on the boat.
Okay.
And we fished for 5 hours, didn't catch a single thing. And TMI, I needed to use the restroom, but was too afraid to ask him to stop because he was tournament fishing and he was intense.
Right.
So that was terrible. I didn't catch a single fish. He didn't catch a single fish. I hated it.
Okay, hold on. I want to pause here because I didn't know we were going to go down this route. But no, I mean, seriously, Cynthia, this is so important. We talk about, like, how do we get more youth anglers involved? How do we get more women involved? Not having a restroom on a boat is kind of it's a barrier. We don't understand it, guys. It's a barrier.
It's a barrier.
I mean, we're okay losing a sock here or there out in the middle of the woods, but it's a barrier.
Yep.
One that we probably need to start thinking of some creative solutions there, boat manufacturers, to help us out.
Yes, please. Because not all women are willing to lean off the side of a boat the way I am now as an adult.
Right.
But a 16-year-old me was not willing to do that.
Especially with a boyfriend, you know, I get it. In tournaments, I get it. But the other one, you know, being 4 years old and having an older brother that can introduce you to fishing. ASA has a great stat that if you're not introduced to fishing by the time you're 14 years old, you won't fish.
Really?
You being introduced by your brother also makes sense because almost every angler, youth angler, is introduced by a male relative. Your brother stepped in and did that for you.
Yep.
And you never know, the point of this, you never know by taking a kid out fishing if you're going to change their entire trajectory of life. And it didn't happen for you for quite a while after that.
Nope.
But once you re-engage with that passion, you now remember these 3 distinct fishing trips, albeit maybe because some horrible things happened in each one.
Because none of them were good.
But you were introduced. And I think, you know, yes, this podcast, if we take an answer away from, like, what is blotchy bass, that's awesome. If we take an answer away from how anglers can help science, that's awesome. But also, take this away. Take a kid fishing.
Yes.
Bailey, don't kidnap a kid, but go grab a kid and take him fishing.
And there are tons of fishing groups actually in DFW. There are several fishing groups that their primary goal is to get people fishing. Like they're a huge Facebook presence. They welcome everyone from all walks of life. You don't have any fishing gear, but you want to try it out? Give us a call. We'll loan you some equipment.
Right.
If we have it, we'll give you the equipment.
Love that.
To get them started. I can't tell you how many kids these anglers have been able to introduce to the sport that we know and love.
Right. Okay, now, true or false? You dumped the tournament bass fisherman because he skunked that day.
Yeah, true. True. No, because actually my third fishing trip was also with him.
All right, tell us how horrible that one was.
Went to a pay lake. In Texas, they call them stock tanks.
Yep.
But in Tennessee, they were pay lakes.
Okay.
And go Vols. We went to a pay lake, and you were allowed 5 fishing rods per person. So there were 2 of us, so we had 10 fishing rods out on the shoreline, which was ridiculous.
That sounds
like
ridiculous.
I do not promote this whatsoever, but this was back in the late 90s, early 2000s. So
you don't have to say that. It's okay.
Before people were kind and courteous, but we get out there and he's rigging up all the rods. Rod number 5 catches a catfish.
Right.
And he says, "Get it, get it, get it." So I get it and I attempt a hook set, which then crosses 1 through 4. Catfish takes off, crosses 6 through 9, which he has 10 in his hand. tangled up all 9 rods. He almost dumped me for that one.
I can imagine.
So he was angry. We didn't take any catfish home. We were out, the $50 because it was $25 per person, and it was not a quality experience.
Yeah, you had 3 horrible experiences.
Mm-hmm.
Congratulations.
Yes.
But then you get to college, you find your love of fresh water.
Well, actually, I was still in saltwater in undergrad. So at Tennessee Tech University, you were allowed to get a biology degree with a marine sciences concentration.
Right.
And what's crazy, you guys heard me mention Tennessee, Del Hollow Lake. I grew up 20 minutes from Del Hollow Lake, which means I was about 30 minutes from the fish hatchery that is right there.
Absolutely.
That I did not know existed because my school never took a field trip there or anything like that. No offense to my school, that just, they didn't know how to guide me. So when I get to college, my undergraduate professor, Dr. Brad Cook, never tried to steer me to freshwater. He was just like, she wants saltwater, and he did everything he could to help me flourish in that field. So as part of the biology marine science concentration program, I had to go to a coastal campus and take some courses in marine biology.
That makes sense, yeah.
And so I went to Gulf Coast Research Laboratory, which is the satellite campus of the University of Southern Mississippi in Ocean Springs, Mississippi. Mm-hmm. And that was the best summer of my life to that point.
Right.
Unfortunately, the coast had just been hit by Hurricane Katrina, and the campus was hit very hard, and they lost their entire specimen library. But silver lining, my class got to go out and collect all new specimens.
That's amazing.
And so we caught tons of sharks and fish and rays and jellyfish. So many things. I got to see so many. I saw a 10-foot tiger shark.
Right.
A 10-foot tiger shark. Granted, we didn't pull it on the boat because it was far too big, and it wouldn't have been safe. But I got to see all of that. And I actually got to reel in my first fish, which was a 3-foot Atlantic shark-nose shark.
Okay.
And I was ecstatic. I was like, yes.
So your first successful fishing trip was actually a coastal trip collecting specimen in college as undergrad. Amazing.
Yes. And so then I get to my master's degree in Louisiana, where I got to do both coastal and saltwater. Okay. I'm sorry, coastal and inland fishing. Yep. But I still enjoyed the coastal fish the best. Red drum, phenomenal.
Right.
And so I still was like, I kind of want to do that. But then I loved the management. I wanted to be able to work with kids, and you're going to be able to do that the most with freshwater. So I stuck out with freshwater. Okay. Loved my project. Got to talk about a fish that a lot of people didn't know much about. It was the yellow bass. There hadn't been a lot of research done on it. So I'm a little bit of a yellow bass expert, at least for the South. And then I started my PhD project at Mississippi State University, where I got to study largemouth bass on the island of Puerto Rico.
Yes.
How does that work?
With one of the amazing researchers,
Dr. Wes Neal.
Dr. Wes Neal. Yeah.
Yes.
So tell us about Puerto Rico. Bass aren't native there, right?
They are not. They were introduced by Puerto Rico Department of Natural and Environmental Resources. And going back to the great debate about Florida versus Northern, they thought they were introducing pure Florida bass. Dr. Wes Neal's lab was actually able to determine that they weren't pure Floridas, unfortunately, and so they are all intergrades on the entire island. But it was Dr. Wes Neal who was actually able to figure out that the main reason that I didn't like fishing, because I never did it recreationally while I was in college. I only did it as part of courses. He's like, you don't dislike fishing, you don't know how to fish.
It's a good clarification.
And I said, okay. So I had just won a speaking competition, so I had a little cash prize with that. And he said, let's go to Bass Pro, let's rig you up. And then he and my lab mates all went to Pickwick Lake.
Okay.
I caught 15 different species in 3 days.
Yeah.
Best fishing trip ever.
Which, for all of us, we're like, yeah, but how many bass did you catch?
You say
Me and
that.
Bailey are like, yeah, okay, 15 species, but what about the bass? Two
Yeah.
bass.
But also because I was very new, I was a novice. I was fishing with very small tackle,
Okay.
lightweight tackle,
Yeah.
so that I would have a better opportunity
Right.
of catching more fish.
And you caught crappie?
Caught crappie, caught some skipjack, caught bowfin,
Yeah. Oh wow.
shad. Gosh, I felt like it was everything in the lake.
Amazing.
It was so much fun.
So we owe you being here in Texas and working so hard to make our inland fisheries better to Dr. Wesley.
Pretty much.
Yeah, I love that.
Yeah. I mean, I hate to give him that much credit.
No,
I'm just
he'll
kidding.
clip this himself and he'll have it. Yeah, and he's an amazing artist. And
Yes. I don't even know if the man manages fish anymore because his art is so fantastic.
Yep.
It's
So
amazing.
let's go back to Puerto Rico.
Yes.
Because I think our audience, some of us would like to travel to Puerto Rico. I'm going next year, so I want some tips.
What are you planning to fish for?
I'm here to ask.
Okay. Well,
I'm gonna catch bass though.
Well, are you going to catch largemouth? Or are you going to catch peacock? Because there are peacock bass on the island.
Great question. School me.
Okay,
Tell me what I need to know.
So some reservoirs only have largemouth. Some reservoirs have both largemouth and peacock.
Okay.
Because thanks to the aquarium trade, several cichlid species, firehead cichlid, guapote tigre, with jaguar guapote is the English translation of guapote tigre. Beautiful fish, but they are cichlids, so they grow quickly, and they often will grow too fast for largemouth bass to be able to eat them. So they outgrow the gape width of largemouth.
Yep.
So peacock bass were introduced because they have a larger gape width than largemouth bass.
To control it.
To try to control them.
Gotcha.
It's not been very successful, but it has led to some phenomenal fisheries.
Okay.
Because it's really awesome to be fishing for largemouth bass, and all of a sudden you get this bright orange, yellow, and green behemoth on the end of your line that comes up, and it has the big nodule like cichlids do. And I highly recommend you go to Oaxaca. It's going to be on the west side of the island.
Okay.
That's where you want to go, because that's where you're going to get good largemouth bass and good peacock bass.
Okay. And do I need to hire a guide, or can I just run a boat?
You can just run a boat, but there are plenty of guides specifically for Oaxaca, because it is one of the most popular fisheries on the island, or at least it was 10 years ago when I was there. It may have changed since then, because they have unfortunately been hit by hurricanes a couple of times, but that actually did lead to damaging the dam and the spillway at Oaxaca. So I don't know if the fishery has rebounded, but when I was there, that was where you wanted to go. And in fact, my dissertation project, we are all familiar with bass crowding or stunted bass growth. It doesn't typically happen in tropical and subtropical systems because they will grow really fast, they get big really fast, they expend all their energy, and they die pretty quickly, normally less than 6 years. But in one particular reservoir, which I will butcher the name because I'm terrible at rolling the R's, but Cerrillos Reservoir is in the center of the island. It's just north of the city of Ponce. It's about a little over 500 acres. All the reservoirs there are municipal water sources. So there's no swimming, and management practices are limited to what you can do without chemicals. They will not allow chemicals in their water. Perfectly fine with that. But in this one particular reservoir, it's unlike most of the others. It is very deep. It's over 80 feet in some areas. And it's oligotrophic, so not very productive except in the upper reaches near the river. But it had a stunted bass population, or bass crowding, which normally we would think, okay, it's a forage issue. So let's get out there and see what we can do about that. It had tons of forage. It had 2 species of sunfish plus threadfin shad. Should not, and 2 species of tilapia. So should not have been a problem. But what we saw is the fish would go really quickly up to about 10 to 12 inches, and then the growth would just stop. You would have a few fish that would grow out of that bottleneck, but very few fish were above quality size. Very few. So we used a study by David Willis from South Dakota, and where they had done annual removals of a certain biomass And then they followed the population to see how it responded, to see if they could change the size structure. We tried doing that, and we hypothesized that it would happen faster for us than it did in South Dakota because they don't really have a winter. And we were correct. We removed, so the bass population there when we started, their relative weight or their plumpness was at about 80, whatever the unit of relative weight is. Optimum, of course, is 100.
Right.
So we decided, just theoretically, okay, let's remove 20% of the population, the whole population, not just the crowded.
How did y'all determine the population size, the number of fish?
So we did a standard tag recapture, mark recapture study for population assessment. And then within that, we found where the most crowded fish were. So fish within the 10 to 14 inch range were the fish that got removed. We did remove a few up to 16 inches when we would catch them, but it wasn't very often that we would. So basically 10 to 18 inches is what we were willing to remove, but most of them were 10 to 14 inches that got removed.
So remove 20% of the population via electrofishing?
Via electrofishing.
Electrofishing.
And then we did not sacrifice them. We actually hauled them to another reservoir to see how they would do. And particularly we took them to Oaxaca Reservoir because the peacock bass had overwhelmed the largemouth bass population. So we were trying to get the largemouth reestablished there. It was successful. But we removed 20% of the total biomass of the population for 2 years in a row. And then we assessed the population through that time. And we were able to increase their relative weight value from 83 to 90 in those 2 years. So then we stopped because that's what Willis and them did. And within 1 year, the population reverted back to where it was.
Well, this wouldn't be an episode of Real Biology if we didn't talk about the harvest of bass. So I love that you had this path in your career whenever you were, getting your PhD, that you actually did this. And let's think about it for a second. 500-acre reservoir, remove 20% of a population, see a 7 to 10% increase over 2 years. The next year, stop doing that, and it reverts back. And the natural harvest by anglers in Puerto Rico is fairly low.
Very low.
Okay, so it's kind of mimics what we see here in Texas and what we see across the U.S., which is angler culture has declined to harvest fish. We are not a consumptive population anymore, so our angler group lets fish go. By letting fish go, they are not going to grow bigger in most scenarios. You don't have to say this. I'm saying this. It's okay. They're not going to grow bigger in most scenarios without some sort of corrective measures, either more forage, better water quality, more habitat, or harvest. In a lot of cases, it's all of those things.
Yes.
And it doesn't matter if they're northerns, intergrades, or Floridas. Genetics can only showcase what the fishery will produce if everything's optimal. So you proved that. You proved a very similar concept to that in Puerto Rico. And it's amazing to me because it's like every guest, we didn't talk about this beforehand.
No, we did not.
Bailey, we didn't talk about this beforehand.
I did not know I was going to be talking about Puerto Rico today.
We did not. But every guest has this similar conversation about their reservoirs, either the ones they're currently working on or in the past, of if we could just get anglers to harvest, the fishery would improve. So theoretically, you guys harvested 20% and saw a 7% increase over 2 years. At some point, that's going to level out. It's not just going to be 7% better every 2 years. But if anglers would have participated and maintained a 10, 20% harvest across the reservoir, and you knew what that number was, how many fish needed to come out, a quota per year, the fisheries would have continued to improve.
Possibly. I hope.
Yeah, you would hope. That would be the theory, right? But we saw a decline within that first year of stopping. So at this point, it wouldn't be an episode of Real Biology if we didn't mention the need to harvest largemouth bass. And I think this is so cool because every single guest we have comes on the show and talks about some point in their career when harvest is so important to improving a fishery. And what you just said about your time in Puerto Rico and removing 20% of this population, 500-acre reservoir, showcases the fact that today we really don't harvest at a rate like we used to. So our anglers aren't participating in making the fisheries better in that aspect of it.
Right.
Because we know we can do things like add forage, add habitat, like you mentioned, water quality improvement, especially on the municipal side.
Mm-hmm.
But that angler harvest component, the reason we have regulations is because we're trying to get fish out of the system because
Yes.
it does improve everything. And these are my words; they don't have to be your words.
Well,
Cynthia.
I have a good example for you.
Okay,
so
go
this
for it.
this is the example that I used in Puerto Rico to anglers there. At least the ones that I could speak to. I would talk to them about Thanksgiving dinner.
Okay.
So if you invite 10 people to your house and you have a 10-pound turkey,
Right.
then everybody gets 1 pound of turkey.
Right.
But if you only invite 5 people, then everybody gets 2 pounds of turkey. And
Okay.
so when I would talk to them about the bass, I said, yes, I completely understand the mentality of, well, if I put it back, it'll grow bigger. But there's only so much forage that a water
well
body
said.
can hold. So if you take a few bass out, then you're leaving more forage available
Right.
to the remaining bass.
Makes
And
perfect
at
sense.
first, they hated the idea because their catch rates at this reservoir had been astronomical. Even electrofishing, we caught 100, 150 bass an hour.
Right.
But when they started fishing, because this was one of their favorite tournament lakes, they always caught large numbers, but they never caught large fish.
Got it.
Well, then after my study, when they started seeing on average larger fish, more healthy fish, they loved it.
I bet.
And so then
Yes.
they were willing to harvest a few fish. And sometimes they would donate them to shelters or to schools or whoever they could send them to. And sometimes they would eat them themselves. There was very little waste is what I'm trying to get at, is they were actually utilizing the fish and then helping to develop their own quality fishery.
Yep. That is the example. You prove it in research. You got it anecdotal. Like, how do I get it through the English brains? That's exactly right. That's perfect.
I think about food. Everything surrounds food.
Love it. Bailey, Cynthia and I are about to have to go teach students, the next generation, hopefully, of fisheries biologists. Do you have anything else for her?
At this moment, no. I think you've answered a lot of the angler theories over the years of multiple things. From, I can't say how many times, but like I'll be fishing with somebody, or you see it on a video, and they're red lips. See that? They're eating crawfish. Got to be throwing a jig or something along the lines of that. Thank you for solving all these quote unquote angler theories. And
well, I did not solve that. That was all soon to be Dr. Clay Rains. He's going to be defending his dissertation later this year, and I know he's going to pass with flying colors. And of course, this wouldn't be possible without USGS, the Eastern Ecological Research Center, all of the state agencies that participated in the project, plus of course the anglers. And Bailey, please, Stephen, please put my information in the comments, my email address. And so if anglers from anywhere want to reach out to me, I am all about trying to answer questions. If I don't know the answer, I will try to find it for you. So please reach out to me. I am all about trying to connect anglers with scientists to get answers to their questions.
And Dr. Fox Holt loves these blotchy bass pictures. So
Yes.
keep sending them to that email address. That'll be so much fun. Thank you so much for being on the show.
Thank you. I really enjoyed it.
Yeah, this has been fun. We're going to go teach students. What's your next topic? Do you remember?
Actually, scientific writing. So how to collect data in the fields.
Yeah, and mine is stakeholder meeting, and then I'm going to teach a little bit of flipping and pitching. Correct technique.
Do I get to participate?
Sounds like you need a refresher course.
Sounds like it. Yep.
Yep. We'll have more success, I promise.
Okay.
Yep. Bailey, good to see you, brother.
you as well, my man.
Thanks, Bailey. It was great meeting you.
Appreciate y'all.